Hello everyone, I'm Sarah.
And I'm Andrew.
And we'll be discussing histology, immunofluorescence, and immunohistochemistry. These three testing styles are powerful investigative tools that yield a variety of information regarding the morphology of tissues. A main idea to keep in mind is that different tissues have unique chemistries depending on the type of tissue and the cells present within it. Additionally, individual cells have unique surface chemistries that can vary depending on the state of the cell and its environment. Subsequently, these specific markers provide information that is helpful in diagnosing diseases. The first general test we will explore is histology. Histology is a technique used to examine small samples of cells or tissues under light or electron microscopy. The samples are prepared by sectioning, staining, and mounting onto a microscope slide, and the preparation can be broken down into 5 major steps labeled here: fixing, processing, embedding, sectioning, and staining. First, after the particular sample has been isolated, it is treated with chemical fixatives, like aqueous formaldehyde, which preserve the tissue from degradation. These chemicals aid in cross-linking proteins for stability, denaturing enzymes that could degrade the sample, and sterilizing the sample from any bacterial infection. Processing of the sample, which includes a dehydration step, replaces the water in tissues with paraffin wax in order to solidify the tissue sample. The sample is further placed into a mold with more wax in the embedding step in order to turn the tissue into a hardened block that is easy to cut. During sectioning, a steel knife that is mounted within a microtome is used to cut 4-micron tissue sections, which will later be placed onto microscope slides. The final step, staining, is employed to give both contrast to the tissue as well as highlight particular features of interest. The most common histological stain is hematoxylin and eosin. Hematoxylin is a basic dye that stains nuclei blue due to its affinity for nucleic acid, and eosin is an acidic dye that stains the cytoplasm pink. But wait, we can modify the assay to get more specificity out of our analysis. To do this, let's explore immunofluorescence. Immunofluorescence follows a similar preparation procedure as histology. However, rather than using a chemical stain, this technique uses an antibody that is tagged with a fluorophore, such as fluorescein and rhodamine. These samples can be viewed through fluorescence or confocal microscopy so that fluorescence can be detected with high intracellular detail. Immunofluorescence is useful because it offers a more specific technique for investigating antigen presence, whereas histology only allows a visualization of the cell structure as a whole.
The third technique we'll discuss is immunohistochemistry. This technique is almost the same as immunofluorescence. However, the antibodies used are conjugated with an enzyme like peroxidase rather than a fluorophore. These enzymes can catalyze a color-producing reaction upon addition of a certain substrate, so light microscopy can be used to visualize the samples. Like immunofluorescence, this technique allows for higher selectivity for a target antigen. Discuss measured and controlled parameters. So in all of the tests, the stain or antibody that's used will create a color change or fluorescence that's able to be seen with the appropriate microscope. This measures the presence of a specific target antigen or a phenotype in the sample. For example, in a live dead cellular assay, which you can see here on the left, dead cells will appear with a red fluorescence, while live cells will appear with a green fluorescence. However, in order to verify that the stain is labeling the right target, Control samples with a uniform phenotype must also be tested. So, in the live dead assay, you would test a positive control with all live cells, which you can see here on the right. In the right image on the left, all of the live cells stain all green, or the negative control on the right with all dead cells staining red. And now for the data. Since the goal of these techniques is to allow for visualization of cells and tissues, the data generated is largely qualitative. For instance, the skin sample that you see here clearly distinguishes between the epidermal and the dermal layers, but it really doesn't offer much in the way of quantitative data. Most of the microscopic images offer insight into structure of the samples, which can also help researchers to understand functions and interactions of cells and their cellular components. However, there's also some quantitative data that can be collected from these tests. In some instances, counting for numerical comparisons of labeled cells is necessary, such as in the live dead assay that I just mentioned. Studies have also explored measurements of fluorescence intensity to determine the magnitude of presence of an antigen. Software packages like QuickCal can be used to translate fluorescence intensity into antibodies bound per cell, or ABC. The data here, which was translated from fluorescence intensity, compares antigen prevalence on T lymphocytes in A, B lymphocytes in B, and between lymphocytes, monocytes, and polymorphonuclear cells in C. So this data has a really strong impact in biology. All the nice fluorescent images that we see, well, maybe not fluorescent for all of them, but all of the nice colorful pictures that we see of tissues and cells are generally created with histology. These images give us an insight into the structure of tissues and cells, which can then be interpreted to understand their function. Having the ability to visualize these microscopic structures that can't be seen with the naked eye is critical for us wrapping our minds around these intricate functions of the cells and tissues in our body. So one example of histology in immunology is the visualization of the lymph node and cells within that structure. The picture on the left here shows an entire cross-section of a lymph node, including the capsule, follicles, the parafollicular cortex, and even some germinal centers. Cell-specific stains also allow us to distinguish between cell types so that we can tell where certain types of cells are localizing. The image on the right, which we also saw in Lecture 2, shows the distinct localization of B cells and follicles, while T cells localize to the parafollicular cortex. There are a few engineering aspects that are related to histological techniques. With the high specificity of antibodies for their target antigen, successful stains can sometimes be a challenge to generate. Sometimes other antibodies that are specific for the desired target antigen are also hard to find in nature. For those reasons, engineering antibodies to detect the exact antigen that's intended to be tested is a good way of amplifying the technique's functions. Also, as I mentioned a few times previously, these tests help us to understand some of the functions of cells by analyzing their structure. If a certain cellular function can be determined from a histological test, further tests can then engineer ways to either use or modify that function in some way. Thank you guys.
Thanks for listening.